Hello, everyone. Welcome to Being Patient Brain Talks. I'm Deborah Kahn, founder of Being Patient. Today, we're going to talk about something called glial cells. A lot of you may not know what they are, but they're really important for many different reasons for our brains. Joining me now is Terrence Sejnowski. He's from the Salk Institute, and he's been studying glial cells to understand their function in relation to many different things. So, Dr. Sejnowski, thank you so much for joining us.
Well, I'm very pleased to be here.
Okay, let's just start simple because I think 99.9% of the population probably doesn't understand what a glial cell is. So, can you tell us what exactly is a glial cell?
Well, first of all, the word glia derives from a Greek word meaning glue, and it basically connects different neurons and with the blood vessels, for example. There are three different types. Astrocyte connects the blood vessel to the neurons with nutrients, also important for shaping learning and memory. This is the connections between neurons. Microglia are really interesting because they're mobile. They actually go in between the neurons and they gobble up all kinds of detritus that comes out of the neurons and helps clean up the mess. The Finally, the third type is the oligodendrocytes, and what they do is they wrap around the way the axons that the neurons use to communicate and speed up the signaling. So they have very different functions, but they're all very important.
Okay, let's start with what most people probably may have heard of, which is the microglia. They're known as the janitors of our brain. As I understand it, they really only come out in deep sleep. That's why sleep is so important. Is that true?
No, they're always there. That's to say, you're right. They have an important function during deep slow wave sleep, which is to clear out the space between the neurons. A lot of, for example, especially in Alzheimer's, the plaque, the amyloid beta plaque, has to get disposed of. But no, but they're monitors. They go around, and if there's any damaged part of a neuron or a synapse, a connection between the neurons, they clean it up. And it's a, like you're right, maybe it's a janitor job, but without the mass the microglia, you would have really be in bad shape. You wouldn't be able to, like you said, sleep would really be disrupted.
So I thought though that that's why it's so important to get sleep because in deep sleep, the microglia are actually cleansing our brains, like purging toxins or what's not supposed to be there. And that really only happens at nighttime.
The actual purging. What happens actually is very fascinating. Macon Niedergaard discovered this. What happens is that the space between the neurons expands, and that then allows a flow of material. And the microglia are basically there to make sure that it's going in the right direction, cleaning things out.
Do we know why that happens only in sleep? Do we understand why?
Well, when you go to sleep, your brain, it doesn't stop. It just basically goes into a completely different mode, and it's processing information that occurs during the day to help restore and consolidate the memories, long-term memories, and that's part of the process of cleaning things up that maybe you don't need, things that are very maybe even detrimental, like amyloid beta is that's part of what goes wrong with Alzheimer's, that the cells get filled with it and then they die.
So you have your glial cells and could you say that glial cells are important to the health of neurons and how they communicate, or what role do they actually play in terms of, supporting the structure of the brain?
Well, the more we study them, the more we discover that they're initially they were thought to be nutritive, supportive, and they are. As I said, they carry in essential nutrients into the neurons from the blood vessels, but they do a lot more. For example, when the synapses are talking to each other, There's an astrocyte that is it has all these little arms and it goes in between all the different synapses and it helps those synapses communicate and by exchanging chemicals and they're connected to each other with electrical connections. So they form kind of a structure, a basic structure within which the brain operates. It's really essential.
So, how do we think about these cells in relation to neurodegenerative diseases? Are they being studied to understand really what goes wrong when somebody first shows signs? Maybe when before plaques are forming or while they're forming. Can they offer us any insight into what's going wrong in an Alzheimer's brain, or for that matter, other dementias?
So I have a colleague here, Nicola Allen, who actually studies this, and what neuroscientists are discovering is that they could be involved in the very beginning of Alzheimer's. Alzheimer's, the plaques and tangles, which are seen in the end state when the neurons are dying, occurs way after the early insult that caused it. And there could be many different things that lead to Alzheimer's. It takes decades to develop. Uh, and viruses have been implicated, but there's a line of research that implicates microglia, and they go rogue. Suppose that you have microglia that is starting to eat good synapses and destroy the connections between neurons. I mean, this is worst case scenario, but that does happen.
Yeah, so perhaps maybe you could start even looking earlier if you study these cells. But tell me a little bit about your research, because I know that wasn't directly, but you looked at several regions of the brain and looked at how, the different functions for glial cells. Tell me about your research.
So we had a paper that was recently published in the Proceedings of the National Academy of Sciences Nexus, and it was the work of two postdoctoral fellows in my lab, Shyam Srinivasan, and also who's from India, and Antonio Pino Duarte from Portugal. We have a very international lab. And what they did is to look carefully and counted the number of different types of glia in different brain areas. And they discovered three regularities. We'd like to call them laws, but obviously it's not exactly a law, it's a regularity. And one of the things that was really amazing is that if you look at the density of glial cells, that's the number of glial cells in the volume compared to neurons, it varies across different parts of the brain. It may be more glia in, for example, one part like the neocortex compared to the cerebellum, which is a motor structure, or the olfactory cortex. But however, even though it's different, it's the same ratio. The density is the same across all mammalian species. And that's really quite remarkable. What it says that there's something that's characteristic about the glial cells that makes them the need for them different in different areas.
Do we know about, I mean, like if you drink alcohol, you might be killing brain cells, so to speak, do we know, like, is that a set number in those different regions, or does environmental factors, behavioral factors play into the amount of glial cells that we have? And does that impact things like memory?
Well, first of all, as far as I know, none of the animals that we used in our experiments had any alcohol, so we can't speak to that. However, the fact that it was so universal, I mean, we have about a dozen different species, suggests it is like a law-like, and there must be some reason. We don't know what the reason is. Why is it you need higher density here rather than there? Well, it probably has to do with the function that particular brain area has. Like obviously the olfactory cortex is there to help you smell and recognize smells. Cerebellum is there to help you move. It's part of the motor system. And the other parts of the cortex also have very different functions. So it's really we're actually, this is something in science, is that every time you make a discovery, it opens up a whole set of new questions that you now can study and try to get to the answers to. And so we're this is by the way, glial cells don't get as much publicity, as you probably know, than the neurons. Everybody knows the neurons are important for thinking and for behavior. But I tell you, without the glial cells, they would be in bad shape. They absolutely depend on them. So
Neurons would be in bad shape.
yeah, well, they wouldn't get nutrients. The synapses wouldn't work properly. Things wouldn't get cleared out. Come on, I mean, you need all of that kind of foundational stuff to support the rest of the brain.
So, what does this mean in terms of like how would one be able to, how could we use this as a gauge for brain health, perhaps? So, you know, your findings are, I guess, suggesting that glias may sometimes be a better marker for the brain's region in certain areas than neurons. But what does this mean for, what are those next questions that you're asking in terms of understanding these cells' relationship to, brain function overall?
Well, a couple of things. First, multiple sclerosis is a devastating disease, motor disease. And we now know it's an autoimmune disease, and it's the oligodendrocytes, the ones that are wrapping the axon, get attacked by the immune system. And once you start peeling away the oligos, your ability to walk and your ability to move seriously, disrupted. And so clearly, knowing something about what's going on there and trying to help patients is really important. And another one is the astrocytes I mentioned are important for learning and memory. And let me just give you one experiment that I was particularly struck by. Macon Niedergard, who I mentioned earlier, he's University of Rochester, did the following experiment. She transplanted astrocytes from humans into rats and mice, rodents. And here's what she discovered, that the human astrocytes, which are much bigger than the mouse astrocytes, uh, helped the rats learn faster. Okay, well, that's maybe giving us a hint that these astrocytes are really important for human learning. Uh, and there's a little a humorous story which is amazing. When Einstein died in 1955, his physician kept his brain, and much later it was studied by neuroanatomists because they figured that, look, this is one of the most amazing brains in physics in terms of him being a genius. Let's see which parts of the brain were different. They didn't find any part of the brain that was different in terms of the volume, but what they discovered was that Einstein had more glial cells than a normal person, especially in the part of the brain called the parietal cortex, which is important for your spatial relationships, where you are in space. And he had a wonderful Einstein way of visualizing problems and coming up with solutions, like in relativity. So I really think that understanding those mechanisms that may help others who are having learning problems might be a possible way for us to be able to help people who have learning disabilities.
So, okay, pulling back a little bit, like in terms of what we know about these glial cells, I think I've heard them spoken about in three different groups. It's like, I'm gonna screw up pronunciation, but oligodendrocytes,
That's good.
drocytes,
Yeah. Okay.
astrocytes, and microglia, right? Are
Right.
those the three categories of glial cells?
Those are the three main categories. There's a lot of subcategories. In other words, they're not all the same everywhere, but in terms of the way that neuroscientists categorize them, yes, those are the three main types.
And we know, is it perhaps that we haven't discovered another group of glial cells, or how much do we know today?
Uh, there could well be, and one of the things that came out of the Brain Initiative is a catalog of all the cells in the brain. And there are some new types of cells that no one had ever known about. And as I say, subcategories of glial cells. Now, one of the problems with coming up with identities is that it's not always the case that they're separate, that it could be a continuum, that there may be some in between. And that's really making it more difficult for us to sort of parse out the functions to these different uh, subtypes. But
So is it the relationship though? I think what we're saying is we're trying, I mean, we understand the function. So in some, I don't know if we understand all of their functions, but we understand somewhat, like with microglia and, you were talking about other cases with astrocytes. So is it the relationship between glial cells and neurons that we need to delve into more? Or where's the missing part of the equation in terms of, where are we in terms of, what we need to know?
so you're putting your finger on something really important, and that is the communication between different cells in the brain. Now, I mentioned synapses. That's the a very uh, complex way that neurons talk to each other by releasing a chemical transmitter and then activating the other side, the postsynaptic side. Well, it turns out that glial cells also release these chemicals. In fact, one of the important functions they have is to take up some of the transmitters, glutamate in particular, and then reprocess it and send it back to the presynaptic side so they can be recycled. So it's a recycling system. I mean, it's incredibly important. And what we're discovering is that a lot of the signals that are there are having multiple functions that have to do with regulating learning, how those changes occur in the synapse that make them more strong in terms of their function or sometimes weaker. And there are also, we know, there are something called peptides. These are short chain, like 10 amino acids or less in some cases, uh, that are also released. And the problem is we don't know what those peptides are doing. And so there's a big mystery here, and it's known that the peptides act very slowly, in many cases over many minutes. So they may be having a function in reorganizing the structure of the networks on a longer time scale that may be, again, important for learning and memory, but maybe other functions too. We just don't know.
Right. It's interesting. So would you say, I want to kind of bring it back to neurodegeneration and Alzheimer's disease. I mean, we know, I mean, beta amyloid plaque is the presumed beginning pathology for Alzheimer's in particular. But is there perhaps a hypothesis out there that if we study the way that glial cells, maybe I don't know if misfunction is the right way to say it, but if there is a problem with the glial cells prior to amyloid, then is that maybe one way where we should look at neurodegeneration? Like maybe they can unlock some of the clues in terms of what goes wrong in an Alzheimer's brain. Is that one way to put it?
That's one hypothesis. And that's a hypothesis that may be involved in the early triggering. But there is another hypothesis, which is that the microglia are there doing their best. Like they're just working full time. And by the way, the amyloid beta, there's been 100 clinical trials, phase 3 clinical trials, each with costing hundreds of millions, in some case $1 billion. They've all failed. In other words, just clearing out the amyloid beta is not the solution. The real problem is how to go back and figure out what started it. And I gave two examples. The microglia might be involved; they may go berserk. Uh, there's some evidence for that, but also there's more and more evidence that the viruses get in, and that triggers a bunch of changes that then lead to this very end state, which is the plaques and tangles. This is an embarrassment that we have. It's taken us so long. We put so much emphasis on. It's a marker of the brain at the very end state. And there was a big study that was done of people who have lived 100 years, and some of them had amyloid plaques, uh, and they were perfectly normally functioning. In other words, it's not 100% that amyloid plaques means you have Alzheimer's. So that throws a little bit of doubt on the simple minded idea that this one particular problem of polymerization of a molecule that makes these tangles. That's not the only problem. We're and NIH now is actually looking for other ways to think about how to attack the problem. It's a really it's getting worse because the population is aging, and it's a burden not just on the cost of medical care, but it's a burden on the families. They're suffering too because they have to, it's like 24/7. You have to be the loved ones, right? You have to really care for them.
Yeah, and I mean, what you're bringing up too is the microbe theory, right? Where bacteria and viruses could play a role. I mean, we know with Rudy Tanzi, I think, was researching the herpes virus in the brain.
Right.
So there's a lot more research, and there's not, yeah. So it's encouraging to see that finally science is looking in other areas as well, right? We really don't
Right.
know.
It,
So.
it, it takes time and a lot of work. Let me give you one example. Cancer, right? War on cancer in the 60s. Uh, what they discovered was that cancer was a genetic disease. They discovered that it was heterogeneous, different cell types that became cancerous in different organs. It was different. Each one had to be treated as a separate cancer. And it took 50 years. They realized they're going to have to go down to ABCs and figure out what genes and proteins are involved and do the cell biology. And finally, we're at a point now where we're able to actually cure a small number of really terrible cancers like melanomas, which were death sentences. And now people are living that when they would have been dead if we didn't have the therapy. But it takes 50 years of research, and you have to really get fundamental basic science done so we can understand exactly what the pathways are and identify the targets and develop new drugs. And that is really what we're in the middle of right now. We're just in a sense, we're beginning all over again.
Yeah, and just to bring it back to your research, now that you have studied glial cells and realized that across different mammal species we have the same amounts in different parts of the brain, what's the next step to your research?
Well, the next step is going to be to try to manipulate. What you can do now with modern genetic techniques, you can manipulate genes, and we can identify genes that may affect the density of these different glial cells. And then by looking at the impact on behavior and health, that'll give some hints about what might be their function.
That might make us all smarter.
Yeah, well, I'm hoping for super large glial cells that can
Superhuman glial
for
cells.
humans, yeah, that will make us smarter. But that's not the main goal. The main goal is to actually help people who have problems with their glial cells.
Yeah, well, maybe it's the answer to AI, like supercharged glial cells that make humans smarter, and we're gonna beat the machine.
Well, actually, I'm an expert on something called neuro AI, which is the border, neuroscience now talks to people in AI because they're dealing with the same models, the architectures, a lot of neural networks. Uh, and one of the things that we can do is learn from AI, and AI can learn from us. And I think that AI needs glial cells.
Whoa. Okay, we're coming back to you and maybe how long do you need? 10 years before we can.
No, no, this we're talking about, this will happen within the next couple of years.
Okay, we're coming back in 2 years. That's whoa, that sounds so sci-fi, but really, really interesting. Terrence Sejnowski, thank you so much for joining us. A really interesting story on glial cells. Again, putting the pieces of the puzzle together. We really appreciate researchers such as yourself coming on and letting us know what's going on. So thank you.
Well, you're very welcome, and I appreciate having this opportunity.
Okay, and so if you want to know more about Dr. Sejnowski's research, he's at the Salk Institute. We can post a link to his research in this chat. Don't forget, everyone, to sign up to our newsletter. That's where you hear about talks like these. Thanks, everyone, for watching, and have a great weekend.